author and genealogist Donald Yates, who not only through his own Cherokee ancestry discovered his own Jewish roots, but he's also leading the charge in the DNA discoveries of the ancient roots of the Cherokee and other Native American communities.
In their ongoing efforts to preserve their language, the Cherokee Nation is making a concerted effort to improve and protect the health of its elderly native speakers.
For 100 years, scientists placed the Cherokee in the southern Appalachians and called it settled. Then Haplogroup X showed up in the data, and it had no business being there. New DNA tests finally solved the Cherokee people mystery. The results are shocking. The mitochondrial evidence didn't just point north. It pointed to the exact same coordinates that Cherokee oral tradition had been pointing to all along. What Western science took 3,500 years to measure, the Cherokee already knew. The collision. Here is what scientists believed for most of the 20th century.
I discovered the original Cherokee origin narrative, which was recited at every green corn festival. All the young people were made to learn it in the olden days.
The Cherokee were a southeastern people. Their roots were in these mountains, the river valleys of western North Carolina, the high plateau of eastern Tennessee, the upper drainages of northern Georgia. They had been here, so the thinking went, since deep prehistory. The Appalachians were not just their homeland, they were their origin. The problem had been visible for over a century, and everyone had chosen to look past it. Cherokee is an Iroquoian language. The rest of the Iroquoian family, the Mohawk, the Seneca, the Oneida, the Tuscarora, the Cayuga, all lived in a tight corridor stretching from the Great Lakes through upstate New York to the St. Lawrence Valley. That corridor sits more than 800 miles north of Cherokee territory. Every other Iroquoian language belonged to the northeast, and the Cherokee sat alone at the bottom of the map, speaking a tongue that had no geographic business being there. The gap was not small, it was not ambiguous. 800 miles of Algonquian and Muskogean territory separated the Cherokee from their linguistic relatives.
Many Cherokee children from around here are now taught Cherokee as young as 6 weeks old.
No other Iroquoian people lived anywhere near the Appalachians. The Cherokee were, linguistically speaking, marooned in the wrong hemisphere of the continent. Linguists noticed this in the 1800s and did what scientists do when a problem has no tools to solve it. They named it, dated it roughly, and filed it. They estimated a divergence date, roughly 3,500 to 4,000 years, placing the Cherokee split from the northern Iroquoian groups in the late Archaic period. They said, probably a prehistoric migration, ancient, maybe unrecoverable. The details didn't matter because nothing could recover them. Archaeology could map pottery styles and village layouts. It could not tell you which direction the migration went, who drove it, in what numbers, or what genetic signature it left behind. The question had no answer, until it did. Then the DNA data arrived, and it did not simplify things. It blew the old model apart, the fifth lineage that broke the framework. When geneticists first began cataloging Native American mitochondrial DNA in the late 20th century, the framework they built was clean, almost elegant. 4 founding haplogroups, labeled A, B, C, and D. Each traceable to East Asian source populations, each consistent with a migration across the Bering Land Bridge during the last Ice Age. Roughly 97% of all Native American maternal lineages fit inside those 4 categories.
And it was the story of the Cherokee migration across the Pacific Ocean. So, that began to tell me that the Cherokee have a lot to them that people are not aware of, and that's certainly true.
Papers were published, textbooks were written. The model worked, the confidence was justified, to a point. A, B, C, and D told a coherent story. They had precise counterparts in Siberian populations. They showed the expected patterns of geographic spread. They confirmed what physical anthropology and archaeology had already suggested. 4 lineages, 1 crossing, 1 founding population. Clean, the other 3% did not. A 5th lineage kept appearing in the data, Haplogroup X, and Haplogroup X broke every rule the other 4 lineages followed. Haplogroups A through D all have direct parallels in East Asian populations. Exactly what you expect from a Siberian crossing through Beringia. That is the story the data was supposed to tell. But Haplogroup X, the Native American version, is not concentrated in East Asia at all. Its closest relatives are in Europe and Western Asia, the Middle East, the Caucasus. Populations with no obvious connection to the Beringian crossing in the traditional migration model. Scientists stared at this and did not have a clean answer. This is the exact moment where the story gets hijacked. So I want to be precise about what the data actually shows. Every few years, someone uses Haplogroup X to argue that Native Americans are secretly European, or that ancient Israelites colonized the Americas, or some other pseudoscientific fantasy built specifically to undermine indigenous identity. These claims circulate because Haplogroup X sounds like a mystery, and mystery-shaped data is easy to misrepresent. But the misrepresentation requires ignoring the single most important number in the dataset.
More real people who proved the geneticists wrong. The skinny on that is really the Indians came from Europe. Their mitochondrial types are all Old World from Europe.
Here is what the data shows. The Native American X lineage, designated X2A, diverged from its Eurasian counterpart at least 15,000 years ago. That date is the critical fact. It means X2A was already a fully distinct and independent lineage before anyone set foot in the Americas. It did not arrive through post-Columbian contact. It did not arrive through some mystery Atlantic crossing in the ancient past. It arrived with the original founders, the same people who crossed Beringia during the Ice Age. Haplogroup X is indigenous. Full stop. The lineage traces a branch of the ancient human family tree that spread across Eurasia tens of thousands of years ago. The European connection is deep human prehistory. It tells you about migrations that happened before agriculture, before writing, before any civilization that appears in any history book. It is not evidence of contact, colonization, or anything that happened in the last 10,000 years. That clarification matters because the scientific story of Haplogroup X is genuinely remarkable on its own terms. And that story is specifically about the Cherokee. You don't need pseudoscience to make this interesting. The real data is more interesting than the fabrications. But here is where Haplogroup X stops being an anomaly and starts being a key. Because its distribution was not random. It was not scattered uniformly across the continent in a way that could be dismissed as genetic noise. It clustered, and where it clustered told a story. In 1999, two independent research teams published landmark studies almost simultaneously. Michael Brown and colleagues at Emory University, David Glenn Smith's team at the University of California, Davis. Both groups had spent years tracking Haplogroup X across Native American populations. Both reached the same conclusion. Haplogroup X does not distribute evenly across the continent. It concentrates in a specific region. Among specific peoples, the Great Lakes, Algonquian and Iroquoian-speaking populations.
We want to make sure that that history is shared, that the difficulties of tribal nations and some of the things that have occurred where land has been taken away.
The Ojibwe, the Sioux, the Nez Perce. It appears at notable frequencies across the entire northeastern corridor, and it shows up at lower but measurable frequencies in one specific southeastern population, the Cherokee. The same geographic footprint the language had pointed to for over a century. Two completely independent lines of evidence, genetics and linguistics, both pointing at the same location. Researchers who had spent years on each of those fields separately were now looking at results that agreed with each other. That kind of convergence is not routine in archaeology or population genetics. When it happens, it means something significant is being measured. And there was still a third line of evidence nobody had formally assembled yet. The continent organizes itself. 3 years after those Haplogroup X papers, a researcher named Ripan Malhi at the University of California, Davis assembled the largest mitochondrial DNA dataset anyone had put together from native North American populations. 1,612 samples, 395 full mitochondrial sequences, groups spanning the Arctic to the Gulf Coast. Not a regional snapshot, but a continental map. When his team ran spatial, autocorrelation analyses, mapping haplogroup frequencies against geography and letting the data self-organize, the continent resolved into distinct genetic clusters. Haplogroup A populations had expanded in the Arctic North, Haplogroup B had expanded in the southwest.
And they sailed to cross the Indian Ocean, and they took the northern route, and they established the international dateline, which is essentially where it still is.
Haplogroup X had expanded around the Great Lakes. Now overlay a second map, the known distribution of Iroquoian languages before European contact. The overlap is not approximate, it is precise. The Haplogroup X concentration and the Iroquoian language distribution sit on top of each other like two transparencies showing the same image. And here is the piece that ties the whole picture together. The distribution of Haplogroup X aligns exactly with where linguists place proto-Iroquoian, the ancestral language of the entire Iroquoian family, including Cherokee, before the Cherokee separated and moved south. The Great Lakes region, the Northeast Corridor, the territory that Cherokee oral traditions have described as the original homeland, the place before the long journey to the mountains. Linguistics, genetics, oral tradition, three independent lines of evidence converging on the same map coordinates, not approximately, not arguably, precisely. The old model, Cherokee as an in-place southeastern people with purely local genetic and cultural roots, could not survive that convergence, and it didn't. The data retired it. What replaced it is a picture of a people who knew exactly where they came from, carried that knowledge for thousands of years, and were finally confirmed by the science that had spent a century insisting otherwise. Relatives nobody expected. Then, in 2011, the most unexpected confirmation came from a direction nobody had been looking. Researchers Deborah Lawrence and Brian Kemp at Washington State University had access to skeletal remains from 3 Arikara village sites along the Missouri River in South Dakota. These sites had been occupied between 1600 and 1832. The Arikara speak a Caddoan language, a completely different family from Iroquoian, completely different from Siouan.
In fact, the name for America was the White Land. Appalachian Mountains means the White Mountains in Egyptian. Essentially, they originated as Greeks and Egyptians and Libyan Jews.
On paper, there was no reason to expect any genetic connection to the Cherokee. The Arikara lived on the northern plains, hundreds of miles from any Iroquoian-speaking group. Different language, different ecology, different history, no obvious relationship. The research team was not looking for a Cherokee connection. They were studying Arikara population history on the northern plains. What they found was a result that nobody on the team had predicted and that nobody in the field had anticipated. It stopped people cold. Lawrence and Kemp extracted mitochondrial DNA from 63 skeletal samples. 55 yielded usable sequences, an 87% success rate, which speaks to the exceptional preservation conditions in the Missouri River sediment. The 4 major haplogroups appeared at expected frequencies, but the frequencies were not what made the study significant. What mattered was what happened when the team ran principal coordinates analysis. This statistical method places populations in multidimensional space based on the similarity of their genetic profiles. Think of it as a room where every population is a data point. Points that cluster together share more ancient ancestry. Points that sit apart share less. The Pawnee lived nearby. The Mandan shared the same river. On any geographic or cultural map, those are the populations you would expect the Arikara to resemble most closely. The genetics produced something else entirely. The Arikara did not cluster near the Pawnee, their closest geographic and cultural neighbors on the plains. They did not cluster near the Mandan, who occupied the same river system. Those were the populations you would predict. The genetics refused to cooperate. The Arikara's deepest genetic affinities placed them closest to 2 specific populations, the Siouan-speaking peoples and the Cherokee, hundreds of miles away, Iroquoian language, Southern Appalachians. There is a long-standing and controversial hypothesis in linguistics called the macro-Iroquoian theory. It argues that Iroquoian, Siouan, and Caddoan languages all share a common ancestor so ancient it predates the recognizable divergence of any of those 3 families. Most linguists had considered it speculative. The argument required positing a linguistic relationship so deep that the shared features had been eroded almost entirely by time. Unconvincing, the mainstream said. Set it aside. After Lawrence and Kemp's genetic results, setting it aside became considerably harder to justify. 3 language families, classified as unrelated, separated by hundreds of miles, and the maternal DNA of their speaker populations was pointing toward shared ancestry so deep it predated the languages themselves. The genetic record was pointing toward connections the historical and linguistic record had completely lost track of. If this story is already rewriting what you thought you knew, subscribe. We go this deep every week. What the evidence actually proves. Before the results, what did the dominant model predict? It predicted that Cherokee genetics would look like a southeastern population, lineages shared with neighboring Muscogin and Sioux peoples to the south and west, with diversity patterns reflecting thousands of years of in-place development in the Appalachian highlands. It predicted no strong connection to the Great Lakes, no resonance with plains populations, no genetic echo of an 800-mile northward gaze. The data produced none of that. Cherokee maternal lineages carry clear genetic signatures of a northern origin, specifically consistent with a population centered in the Great Lakes corridor that migrated southward into the Appalachians. The haplogroup diversity patterns, particularly the presence of X2A, and the deep lineage sharing with Great Lakes and Northern Plains populations, cannot be generated by an in-place southeastern people. A population cannot produce that signal by staying put for thousands of years. The data requires movement. A substantial organized population migration into the southern Appalachians during the late Archaic period, roughly 3,000 to 4,000 years ago. This was not a gradual drift. It was not a handful of families trickling south over centuries, a migration large enough to establish a new language as the dominant tongue of an entire region, and to leave a genetic footprint that persists in mitochondrial data 3 millennia later, represents a significant demographic event. The Cherokee arrival reshaped the cultural landscape of the Southeast. It happened long before Mississippian civilization arose, long before the great mound builder complexes that would define the region in later centuries, long before any event recorded in European accounts. This is deep history, and the DNA reached into it. One honest caveat, stated once and precisely: mitochondrial DNA is maternal only, a single line of descent through mothers. It cannot tell the complete genetic story. Modern Cherokee populations in Oklahoma also carry the distorting effects of the Trail of Tears, which killed approximately 1 in 4 Cherokee during the forced relocation of 1838. Population bottlenecks don't falsify data, but they filter it. The eastern band of Cherokee in North Carolina, descendants of those who stayed, represents a less distorted genetic window, but sampling there has been limited. Ancient DNA from pre-contact archaeological sites in the Cherokee homeland would resolve the remaining questions. The extraction technology now exists. The statistical models are sophisticated enough to distinguish migration from admixture. The answer is within reach, and the Cherokee Nation is building the research partnerships to pursue it, on their terms, with their people directing the questions. What the Cherokee always knew. Right now, somewhere. In the southern Appalachians, there are archaeological sites sitting quietly under centuries of forest soil. They hold the physical record of that migration, the bones of the people who made the journey south, the tools they carried across half a continent, the fire pits of camps made along a route that no written record preserved, but that the DNA is now slowly reconstructing. Those sites exist. The technology to read them, ancient DNA extraction, radiocarbon dating refined to a precision of decades, stable isotope analysis that can trace where a person grew up by reading the chemistry of their bones. All of it exists right now in working laboratories. The Cherokee Nation today has more than 450,000 citizens. It operates its own government, its own court system, its own language revitalization programs, and it is increasingly directing how its own history gets excavated, not as subjects of research designed and run by outside institutions, but as the body deciding which questions are worth asking, which sites are appropriate to study, and what the findings mean. That is a fundamentally different kind of science than the kind that spent the 20th century constructing models without ever consulting the people those models described. The early haplogroup studies didn't ask. The partnerships being built now do. Here is what stays with you when you follow this evidence all the way through. The Cherokee always knew they came from the north. Their Kituwa origin story said so, not as metaphor, not as cosmological abstraction, but as memory. Geographic memory, directional memory. We came from the north, and we traveled south, and the mountains became home. That knowledge was carried across generations of forced removal, broken treaties, deliberate cultural erasure, and systematic pressure to forget everything that made the Cherokee people who they were. The schools that banned the language, the policies designed to sever the connection to history, the pressure to assimilate and leave the old stories behind. The knowledge survived all of it. The oral record held. It took Western science 3,500 years of linguistic puzzles, 2 decades of mitochondrial mapping across hundreds of populations, the invention of ancient DNA extraction from 1,000-year-old skeletal remains, the development of statistical methods that could read migration patterns in the frequency of a genetic marker across a continent. All of that, and what it confirmed was the same answer the Kituwa stories had always contained: north. The people came from the north. The DNA tests did not solve a mystery. They confirmed one that was never actually hidden, only invisible to tools that had not yet been invented. The oral traditions were not folklore. They were not mythology passed down as comfort. They were memory, preserved with extraordinary precision across thousands of years of disruption. And when the mitochondrial data finally came back from the laboratories, it said exactly what the Cherokee had always said. The old model collapsed, the stories held, and the science, when it finally caught up, agreed. What does it mean when the traditions a people carried for centuries turn out to be more precise than the science built to replace them? Drop that in the comments, and subscribe, because next time we follow the DNA somewhere even older.